Hi guys, welcome to my YouTube channel Unique Bio Classes based on NCERT syllabus for PU first and second year. If you like my channel, please subscribe, like and share and hit the bell icon so that you can get notifications as soon as videos are getting uploaded. Okay guys, we shall start today's session. In our today's sessions we are going to discuss about the test cross. Okay, in our previous video we have discussed about monohybrid cross. In that we'll come across test cross, which you will find in your textbook on page number 74. Okay, you will find here somewhere here. Okay, so this test cross is what we shall discuss about that one in detail. Here test cross, what I have given a definition of it as an organism showing the dominant phenotype is crossed with a recessive parent. Okay, dominant and recessive I have discussed in my previous videos. You can go to that videos if you have not seen, and you can go through what actually dominant and recessive are. But here I am going to discuss some more information related to dominant and recessive. We shall see. Okay, once again, if we recall about dominant and recessive. Okay, and recessive. Now dominant is what? Which will express. Recessive is what? Which will get suppressed. Okay, one recessive will get suppressed, dominant will express. Dominant will express when? In any condition. In any condition. Whereas recessive will get suppressed when? In presence of dominant. Okay, when dominant is present, then only recessive will get suppressed. If it is not with the dominant, then it will also express. Okay, what are those conditions? Here homozygous and heterozygous. We have two different types of conditions in dominant. One is homozygous and heterozygous. Homozygous is what? Homo means same. Okay, same type of alleles. Like if we are taking an example of tall and a dwarf, hope you remember. Okay, tall and a dwarf in monohybrid cross, we have used this as an example. Homozygous, in that you will find we have given genotype as capital T for tall. Okay, now this one we call it as homozygous, right? Same homozygous if you write in recessive. Okay, in recessive what you will write, because whenever we are giving capital letters, that is dominant, it will express. Recessive in tall and a dwarf was, that was dwarf, which we gave it as small t. Okay, now recessive is expressing when both are in recessive conditions, that is in homozygous condition. Now dominant will express even in heterozygous condition. What is that heterozygous condition? One capital and one small. In this also dominant will express. Now this phenotype is what? Tall. Its phenotype is what? Tall. Whereas in recessive there is no heterozygous condition. Okay, no heterozygous condition. Okay, there is no something like this capital T small t. Why? Because if capital T is present, then definitely it will express, because it is dominant. It will express as tall, not as a dwarf. So this character, this feature is not present in recessive condition. Okay, it is not there. So dominant will express in any condition, either in homozygous or heterozygous, because these are two different alleles, okay, of a same gene. Here see two similar genes, two similar alleles of a same gene. Here you can find two similar alleles of a same gene. Now recessive, when it will express, when it is both are in recessive condition, then only it will express. If it is in heterozygous condition or homozygous condition, dominant will express. Hope you understood this. Dominant we can get two different types. Recessive will get only one type. Okay. So after the, hope you understood this, we shall proceed further with the test cross. An organism showing the dominant phenotype. One organism is there. You can take an example of a pea plant itself, because we have to choose that as an example in a previous video. So the same example we shall continue. An organism showing the dominant phenotype. So now in pea plant, which was showing the dominant phenotype? That was tall. It is crossed with the recessive parent. Okay, dominant phenotype was crossed with a recessive parent. Now recessive parent, when we are saying recessive as a parent, then its a child must be a dominant, that is its offspring. So its offspring we got F1 individual. That F1 individual was tall, capital T and small t. Hope you remember this. Okay, so for that we call it as an organism showing the dominant phenotype is crossed with a recessive parent, that is called as test cross. Even when this it is, or even you can write the another definition, when an unknown individual, when an F1 individual is crossed with a recessive parent, we call it as test cross. You can write that also. Now, what is the significance of this? The method for analyzing the genotype of unknown individual. Okay, this method, test cross, is helpful to know the genotype of unknown individual. Okay, how it is, how we are going to know about that genotype, I shall show you that also. Example, violet color flower, capital W, is dominant over white color flower. We have used small w. Okay, so about this example also I shall discuss. First, I shall discuss about a test cross in uh, a test cross in a tall and a dwarf plant. Okay, so you can find here this monohybrid cross we have used in our inheritance of one gene. This cross you might be having that, right? So phenotype we gave it as tall and a dwarf. Genotype was capital T, small t, small t. Then we gave gametes. Then we got F1 individual. Okay. Now according to the definition of a test cross, what we have, an organism showing the dominant phenotype is crossed with a recessive parent. This is the dominant phenotype. It is crossed with a recessive parent. We call it as what? We call it as test cross. Okay, now this F1 individual, when it is crossed with a recessive parent, we call it as test cross. Why we are doing this? To know the genotype of unknown individual. How we have to know the genotype of unknown individual? Why we have to know? Already we, after crossing this, we have got that this is the genotype, capital T, small t. We are not having here capital T, capital T. We don't have this. Well, already we have got this one. So then why we have to go for test cross? Now when you are growing these plants are growing where? They are growing in the field. Okay, you are growing, you have to imagine that all this what process we are discussing, that we are doing in the agriculture field, and in the agriculture field you have cross pollinated these two plants, and F1 generation you have got one plant which was growing it as tall. Now after seeing a plant we could not determine what genotype it is having. Is it either it is a tall plant or a dwarf plant? Whichever plant it is, we could not say whether it is a dominant plant, whether it is a recessive plant, what genes it do have, what type of genotype it has, we can't say. Okay, so to know the genotype of unknown individual, if we are doing like this, by crossing this, by writing with a pen on capital T, small t, all this, then you can say that this is the genotype. In an agriculture field, after seeing a plant, we could not say. For that reason we have this test cross, so that what F1 plant we have as a tall plant, okay, as a tall here in this cross, it might be a dwarf or some other plant in some other case. So that plant is crossed with a recessive parent, so that we can know what is its genotype. Okay, so we shall try to cross these two and we shall see what genotypes we are going to get and how we are going to identify. Okay, fine, I shall choose this. Now I shall take F1 individual first. Okay, F1 individual, it is crossed with a recessive parent. Which was that recessive parent? Recessive parent is a dwarf. Okay, dwarf is the recessive parent. F1 individual we got tall, we have got there capital T, small t, or we can have capital T, capital T. Here it is small t, small t. For dwarf we gave it a small t. If you see here, for dwarf we gave it a small t. Right, now this tall F1 individual, as that plant is growing in an agriculture field, we are imagining that then this can be having this genotype or this genotype. So we have to try for both. Okay, well, both the genotype should be tried, then we can conclude then which one is the genotype actual. So I am taking, so tall, if we have got F1 individual as tall, it can have either heterozygous genotype or homozygous genotype. For both we have to cross it. First I shall take this homozygous one. Okay, similar type what we have crossed earlier, we are taking capital T here and small t here. Okay, what you will get? You will get after fertilization, capital T, small t. This is tall. Okay. Now, if you have got here, we will get two different types of genotype. One is capital T, another one is small t. Okay. Now, if we take this one from here, you will get capital T, small t, and another one is small t, small t. Right. So, if you have this F1 individual, okay, F1 individual is this one, then in the offsprings you will find there is a tall and a dwarf character. Okay, if you are getting only a tall character, then it shows, then it is not having this genotype. For how you are going to identify this, we have got from this cross, right? Both are homozygous. If we cross with the heterozygous, we are getting tall and a dwarf. So now with this we can make out that this is the genotype. This must be the genotype of F1 individual. How you are saying this? Because when we are using this one, tall and a dwarf, then in the offsprings we have got both the features, as you can find in the F1 individual here. If this is capital T, small t, then only you have got dwarf character here. If it was capital T, we would have not got this character, right? So when we have got dwarf in the F1 gen, in a F2 generation, then F1 generation must be in a heterozygous condition, not in a homozygous condition. If it would have been homozygous, we have got all tall. If it has been heterozygous, we'll get tall and a dwarf. So in F2 generation, we have got tall and a dwarf. So that's why we can say this F1 individual or unknown individual, it is heterozygous in condition. Hope you understood this. Okay, this is how you are going to cross. This is an easiest method which we have shown both together, but this is not the actual method how you have to cross. Actual method how you have to cross, it has been shown on your textbook. In your textbook on page number 75, this cross as an example. Okay, hope you find this. This is a diagrammatic representation of homozygous of a test cross, and this is an example of violet color flower and a white color flower. Okay, you can find here a dominant phenotype it has been given. Dominant phenotype is of what? That is violet color flower. This violet color flower is there. And now if you see like this, okay, without seeing anything, can you say whether this flower is a homozygous or heterozygous by seeing that one after taking a look of that flower? No, we could not say. After seeing this white flower, can we say that it is a dominant or it is a dominant or recessive? No, we can't say. So how we can say is that we are choosing this plant as we wanted to know this one violet color flower. It is a dominant, and whether it is a homozygous or heterozygous, what is its genotype? If we wanted to know, we can choose both the methods. Okay, here it has been given it as homozygous. White flower is recessive conditions. Here also this white flower, it has been taken it as in recessive conditions. So if I show you that one here, violet color flower, violet color is crossed with white color flower. Now white color flower, it has small w, whereas violet color flower, it can have capital W, one another one is capital W, small w. I shall use something like this so that easily you can understand. Okay, first I shall choose this one, capital W. For this we'll get same type of gametes, something like this. This here also will get a same type of gamete. If you write once, that is also enough. From this, how many gametes will get? Two types of gametes. One is capital W, here one small w. First I shall cross this one. For this you can use a checkerboard, something like this. Here we are going to write, okay, here we are going to write one side male and a female, and I will write these capital W on one column and this small w on another column, another row. Okay, we are going to write the same checkerboard Punnett square what we have used in the monohybrid cross. So now these two, how we are going to write, one is capital W, another one is small w. Now these two, capital W, small w, again capital W, small w, here capital W, small w. Now this one is capital means violet. For violet we have used capital W, so it is violet. Here also violet, violet and violet. How we are writing violet? Because wherever there is a capital W, then capital will express. So violet you have it 4 times. Next, same this cross. If we take this one like this, okay, same, one side female, another side male. Again I will use this capital W and a small w. Here small w, small w, this small w coming from this side. Okay, now if you cross this capital W with the small w, capital W with small w, see here small w, small w, small w, small w. Okay, now what you will write this as violet. Why? Because it consists of capital W. Here this one will be violet. Here what you are going to write, it is white. This one is white. Okay, so now in this, if we have got both the varieties violet and a white, then from where we'll get both the varieties? In heterozygous condition, where we'll get all will same, that is only dominant will express, only dominant color will come when it is in homozygous condition. So normally it depends on, if we are getting all are having dominant feature in the offspring, then the F1 individual are homozygous. If there is a different, both the types dominant and recessive both we can find in the offsprings, then we can say it as recessive, that it is in heterozygous condition. Now how you will write? It's a phenotypic ratio. Okay, phenotypic or genotypic ratio both will remain same in test cross. Okay, phenotypic ratio, if you take it here, right, of heterozygous individual, it will be 2:2, right, 2 violet 2 white. Now in place of that we can give it as 1:1 also. Now genotypic ratio, how you are going to write? It is again 2:2, capital W small w, like that we have 2, and here also small w small w we have 2, so 2:2 we can write it as 1:1. So always in a test cross you will get 1:1. Always in a test cross the ratio will be 1:1 of a monohybrid. If we are choosing dihybrid cross, then it will be 1:1:1:1. Okay, this is how you are going to write. So the same thing you have in your textbook, something like this. They have chosen here capital W crossed with the small w, here capital W small w crossed with the small w, and they have got this one. All flowers are violet, unknown flower is homozygous dominant. Okay, if all flowers are violet in color, all flowers are of one color of a dominant type, then it is the unknown flower is, this flower is homozygous. If all flowers are having, half of the flowers are violet and half of the flowers are white, then unknown flower is heterozygous condition. Okay, this is how you have to cross. Test cross, it has been asked for 1 mark, what is test cross, what is its significance and all, and 2 mark question also it has been asked, so be prepared thoroughly for it. So as I give significance, the method for analyzing, it is a method for testing a genotype of unknown individual. We are going to know the genotype of unknown individual. Okay, so do remember all this. Hope you have got this. With that, I wanted to let you know one more information. With the test cross, you have information regarding by a back cross also. About back cross, it has not been given specifically in your textbook, but you should know it's quite easy. Back cross, the cross of a progeny individual with one of its parents. Okay, so what it is here, progeny individual has been taken, it has been crossed with any one of its parents. One of its parents means what? This is the progeny. Same example I am using. This is the progeny of an individual. If it is crossed with any one of the parent, we call it as back cross. Test cross was what? Test cross was F1 individual crossed with the recessive parent. Only with this parent, if it is crossed, we called it as test cross. If F1 individual is crossed with either recessive or dominant, we call it as back cross. Only that is the difference. Okay, so back cross is the cross of progeny individual with one of its parents. We call it as back cross. So this is about back cross and a test cross. If you have any doubts, any queries, any comments, please comment it in the comment box. I shall try to solve those questions. And please like my channel, please subscribe to my channel, don't forget. And we shall meet in my next video with the dye hybrid cross. Okay, stay safe, stay healthy.